Hi, I'm Mrs. Roberts. Welcome to my science lab, aka my house. I'm here today to show a really fun and cool science experiment demonstration that you can do at your own house. I'm here to show that you can do science anywhere at any time, and honestly with some pretty easy ingredients and materials. So first thing you'll notice is that I've got a lab coat on. Not totally necessary, but it is a cool look. I've also got some goggles. Makes you look like a scientist, makes you look cool, keeps things out of your eyes, but also not totally necessary for this demonstration. I've also got gloves. Also not totally necessary, but if you want to look and feel like a scientist, go for it. Nobody's stopping you. So you'll see that I've got all the ingredients set up today. Today, can you guess what we're doing based on the ingredients? We are going to do my favorite science demonstration. And my favorite science demonstration is extracting DNA from a strawberry. So, something that a lot of people don't know is that plants, or they don't even think of, is that plants are living things. They need water, they grow, they have everything that a living thing needs. They've got cells, they've got DNA, which is really cool, just like people. So, because they have DNA, do you know what DNA is? DNA is a molecule, it's a structure inside of our cells. So they're in human cells, they're in animal cells, they're in plant cells. Basically, DNA makes you who you are. It's the set of instructions, kind of like a blueprint for a house or instructions to build a Lego model. They're very important. They tell you who you are. And so anything with DNA is living. And so I'm going to show you how to get that very important molecule out of a strawberry today. Now you might be asking, why are we using a strawberry? That seems completely random. Well, you could use other things. You could use a kiwi, you could use a banana, you can use a lot of different things. The reason I'm using a strawberry is because, first of all, the cells, the seeds are on the outside of the strawberry, so very easy to extract and get out. But also, strawberries have 8 times the amount of DNA per cell than humans do. So it's very easy to visibly see. So because they have 8 times the amount of DNA, it's jam-packed with DNA in every single cell inside their nucleus. It would be awesome if I taught you about cells sometime, but cells are really cool. So let me tell you what you need. You need 1 or 2 strawberries. You could have more, you could have less. You just need really 1, but I'm going to do 2 just for the sake of the demonstration. You're going to need a Ziploc bag that seals. You're going to need some kind of dish detergent, soap. You are going to need salt, just regular salt. We're going to use a 1/2 cup and a teaspoon measurement. You will also need water. You will need a coffee filter, or you could use a cheesecloth, whatever strains the water out is fine. You will also need some kind of glassware. You could use plastic, but I'm trying to be enviro-friendly, so I'm using, this looks like a beaker, but it's not. It's just a little coffee mug that my cousin gave me. You could use a little glass like this. You could use a mason jar. A lot of people have these just sitting around their house. Super easy to just go ahead and grab. Something else that's really important that you need is 91% rubbing alcohol, and keep it cold. So you're gonna need to put this in the freezer before you actually start. I'll tell you why once we actually start the experiment. And then I brought, you could use a coffee stirrer, you could use whatever. I've just got a chopstick because it's going to be used towards the end once we actually have our DNA. So once you have all your materials, we can go ahead and get started. So go ahead and take your plastic bag. You're gonna open it up, and I'm gonna start with the most fun part. So you're gonna take your strawberry. You can peel off the green part if you want. You don't have to. I'm just gonna leave it on because I'm lazy. So they're in there. Take out all of the air. That's out. First, you're going to have to get the DNA out of the strawberry. So first thing we're gonna do is break it down. So you're just gonna take your fingers, you're gonna squish it. Squish, squish, squish. So you'll notice that once you start squeezing it, I'm using my index finger and my thumb. Once you start squeezing it, then it's gonna break down into little chunks. You'll notice that there's green pieces, there's big chunks of red. Just keep doing that. And it's easier to do it at a table. And if you notice that there's still air in it, just take out the air. So, something I say to my students, I've been teaching for now, I'm going on 8 years of teaching middle school, is don't use your fist and just jam on it. The reason for that is because your bag may pop, and if it pops, you're gonna lose all of this yummy strawberry juice that you just created, and you're gonna have to do it all over again. And first of all, that's a waste of food, but it's also a waste of bag, materials, time, all the things. You get the gist. So you are going to smush it and mush it until you have really no chunks left. You can have a few chunks, especially if you left the green leaves on there, but it should look generally like strawberry mush. So once you have that, go ahead and put it aside. We don't need it right now because next we're going to create something called the extraction buffer. And I know that sounds super fancy. Basically, it's a solution that we are going to put in the bag with the strawberries. It's going to extract the DNA. So it's going to separate the DNA from the strawberry parts that we don't need to use. So to create the extraction buffer, first we are going to take 2 teaspoons of dish detergent. So we're going to go 1, we're going to go 2. There we go. Nice bright blue. I like using bright blue dish detergent just because it looks more sciencey. Next we are going to take our salt. So we need a teaspoon of salt. So I really don't want to dig this in, so I'm gonna pour it in. Oops, close enough. We're gonna use our salt. And then, oh, hold on, I forgot water. Let me go grab water, 1 second. All right, I'm back. So we're also gonna take water. And we need half a cup of water. So I've got my half cup. Also, by the way, you shouldn't really drink lab supplies, but we know it's just water and it wasn't contaminated, so it's okay. So we're gonna take our stir. Stir, stir, stir. You really don't want to get too many bubbles. I kind of did it a little bit aggressively, so you might want to stir it a little bit more calm than I stirred it. You just want to make sure that salt and that dish soap and that water blends pretty evenly together. Now, instead of just salt, soap, and water, this is now your DNA extraction liquid. So fun fact before we continue, DNA is actually short for a longer word called deoxyribonucleic acid. Now I know that's a long word, but a challenge I like to give my students is to say it over and over again and then to tell one person in your life that word. So I'm going to repeat it. DNA is deoxyribonucleic acid. And then try to say it as fast as you can. Deoxyribonucleic acid. So cool. Makes you feel really like a scientist. So now we've got our fancy DNA extraction buffer. We've got our strawberries. We are going to now pour this all into the bag. Mix it up, mix it up, mix it up. Next, very important, is we are going to, I'll go ahead and take a clean cup. I'm going to take my mason jar. I'm going to take one of those filters. Oh, don't fly away, filter. I'm going to take one of these filters and I'm going to gently put it on top of here. You don't want the filter to actually fall into the cup. So what I tell my students and what I like to do is I roll it over the side of the cup. And when it's time to pour it in, you might need a friend or someone at home to do this with you. But what I do is I hold it with one hand and then pour the liquid in with the other. So what we're doing now is we're gonna get out all those solids. So we really don't want any mushed strawberry, we don't want any stems, we want just the DNA, because that's what we're looking to see today. So I'm gonna go ahead and carefully and slowly pour this in. And what you'll start to see, I don't know if you can see it, but that liquid is now filtering through the filter. And that's exactly what you want to see. You don't need a lot of liquid, so I kind of went overboard with 2 strawberries. You can kind of just do 1 and even put half the amount of the extraction buffer. So it's gonna slowly drain. And you also don't want the filter to break. Because if you think about it, if the filter breaks, then all this was for nothing. You just kind of put all this stuff together and it just broke through. So you want to be super careful not to break it. So I'm just gonna kind of let it sit here for a minute. All right, once it's been sitting for a little bit, if you're feeling daring and bold, what you could do is you could pick it up like this and then just gently push on it. I think I've got just about enough. Once I have that, I'm just gonna put it in my disposal, and you'll see that it's just filtered out juice, basically. And now that we have that, we are going to take the rubbing alcohol. Oops, it's covered with ice. We only really need half a cup of this. I'm just gonna kind of pour until I feel content with what it looks like, because what you're gonna have is that the alcohol is less dense than this liquid. And so because it's less dense, there's less molecules built up together. And so it's going to rise to the top of this liquid. And our goal is to see the DNA rise to the top. So let me go ahead and show you how to do this. All right, here we go. And you want to be kind of careful so that you can really see the separation. I'm going to kind of put extra in here to make it really visible. You don't want to disturb the layers. Oh, it's already forming. So the reason that it was cold is because when it's cold, it makes it so that the DNA is insoluble. So it can't dissolve in the water or the alcohol. So because it can't dissolve in the alcohol, it's just going to clump together. If it was warmer, then it's just going to basically break down, and that's not what we want. We want it to clump together so we can see it, because DNA is a double helix. It's basically 2 strands spun around each other, and so it's a clumped up molecule. And so actually we did a really great job, guys. We've got a ton of DNA here. So what you'll see is that on the bottom, we still have that strawberry liquid that was filtered out. And then in this alcohol layer, all the DNA is coming towards the top. And so it's that cloudy material. This is what we're looking for. That's DNA right there. So this is all the cells need inside the strawberry to tell it what to do, which is so cool. Deoxyribonucleic acid, don't forget. Thank you for joining me today at Mrs. Roberts' Science Lab. This was my first video, and I'm really excited to share more science experiments with you. If you could like and subscribe, and let me know what you think of this video, how I can improve. Thanks for joining me today. I hope you had fun. Bye.